Welcome to the Biopharma Business Intelligence Unit, where symbolic integrity meets strategic clarity. At BBIU, we go beyond the headlines to decode the structural forces behind global biopharma, AI, and economic realignment. If you value precision, verification, and symbolic depth, this channel is for you. Subscribe now to access our weekly briefings, dossiers, and intelligence reports, and consider joining our institutional tier to unlock early warning systems, custom metrics, and exclusive strategic insight. You're not just listening, you're entering the future of epistemic intelligence. www.biopharmabusinessintelligenceunit.com.
The role of preclinical phase in drug development, objectives, methods, and strategic risks. Aug 29th 1 Introduction. The preclinical phase of drug development is the structural bridge between discovery and first-in-human trials. While its direct cost is comparatively modest, its epistemic and financial weight is enormous. Failure to detect critical safety signals at this stage can translate into catastrophic losses once a candidate enters clinical phases. 2. Core objectives of the preclinical phase ICH framework. Safety evaluation, acute and repeated dose toxicity studies in 2 species, one rodent, one non-rodent, to identify target organs and determine NOAEL, no observed adverse effect level. Pharmacokinetics and toxicokinetics, METK, characterization of absorption, distribution, metabolism, and excretion, correlation with systemic exposure. Safety pharmacology, cardiovascular, CNS, and respiratory evaluation, ICS-7A, S7B. Reproductive and developmental toxicity, required if women of childbearing potential will be exposed, ICS-5. Carcinogenicity, for chronic use small molecules, ICS-1. Biotechnology products, special requirements for immunogenicity and cross-reactivity, ICS-6. Regulatory integration, all studies documented under GLP and aligned with FR2 to support IND-CTA submission. 3. Methodological core. Histopathology toxicology cannot rely only on biomarkers or clinical signs. Sacrifice of animals at scheduled intervals provides histological samples from all major organs. This remains the gold standard for detecting subclinical lesions, correlating biochemical findings with microscopic damage, and establishing a safe starting dose in humans. Despite progress with organoids and imaging, histology remains the regulator-approved baseline. 4. Y2 species, rodent and non-rodent rodent, rat, mouse, extensive toxicological database, low cost, short life cycle, transgenic models. Non-rodent, dog, monkey, mini pig, larger body size for repeated sampling, metabolism closer to human, essential for biologics where rodent receptors may not be relevant. Together they form a system of epistemic redundancy, 2 mirrors at different angles to reduce translational error. 5. Economic dimensions. Preclinical package cost, USD 1080 member, depending on modality. Clinical phases cost, Phase 1, USD 20 to 50 MIMS, Phase 2, USD 50 to 100, Phase 3, USD 300 MIM to 1B+. If a preclinical failure contaminates data and the candidate advances by error, the loss can escalate from tens of millions to billions. Beyond financial damage, reputational and legal risks can destroy an entire company. 6. Failure and data integrity. Legitimate failure, unexpected toxicity, early kill, relatively low cost. Data contamination, errors in GLP, cross-contamination or fraud, regulatory rejection, forced repetition of studies, collapse of investor trust. The difference is existential. A clean failure is acceptable. A contaminated dataset undermines credibility at its root. 7. BBIU perspective. The preclinical phase is not a bureaucratic box-ticking exercise, but a structural filter of epistemic integrity. When well executed, it saves patients from harm and companies from ruin. When neglected, it generates false safety that magnifies downstream costs exponentially. For investors and policymakers, the robustness of a preclinical package is a direct predictor of survival across the clinical pipeline. 8. Educational takeaway. Medical professionals, regulators, and executives should understand the preclinical phase as more than animal testing. It is the sentry post of drug development, where science, ethics, and finance intersect. Sacrifice of animals for histology is not cruelty but a regulatory necessity until validated alternatives mature. The true ethical risk lies in allowing unsafe drugs to proceed to human trials. AI-based 3D molecular design offers acceleration but cannot bypass the mandatory empirical safeguards defined by ICH. Final note at BBIU. We emphasize that integrity at the preclinical stage is the strongest determinant of long-term success in drug development. What fails here saves billions later. What slips through here can cost everything. Case study, Fialuridine, FIAU, 1993, and the rationale for staggered phase 1 dosing. Fialuridine, a nucleoside analog developed for chronic hepatitis B, advanced to clinical testing after apparently safe preclinical studies in rodents and dogs. Yet, during a phase 2 trial at the NIH in 1993, 5 out of 15 patients died of fulminant hepatic failure caused by previously undetected mitochondrial toxicity. Why this matters for phase 1 design. The FIAU disaster revealed the inherent limits of animal data. Standard species metabolize the drug differently, masking human-specific risks. Since preclinical studies cannot provide absolute certainty, regulators redesigned phase 1 protocols to minimize exposure if hidden toxicity emerges. Escalating doses. Starting from a fraction of the Noel-derived dose, small cohorts are treated sequentially, single ascending dose, multiple ascending dose. This prevents exposing many people to a potentially unsafe level at once. Staggered participants, sentinel dosing. Even within a cohort, the first volunteer is dosed alone, followed by careful observation before others receive the drug. This allows early detection of unexpected reactions before group-wide exposure. Structural lesson. The deaths from Fialuridine and later the TGN-412 cytokine storm in 2006 cemented the principle that phase 1 is not only about testing safety, but about limiting catastrophic risk through stepwise, staggered exposure. What failed in preclinical must not be allowed to harm entire groups in early human trials.
.com